Greetings and welcome to, whoa, didn't mean to go there yet, Chapter 12, DNA Replication Part 1. Getting a little laser pointer going here. This is one of 4 video lectures. I know you're excited about that. Topoisomerase. Word about topoisomerase. It is quite the word. Replication and cancer. So here are the leaves of a tree known as the happy tree. And from the leaves of this tree we get a chemical called camptothecin that was used successfully to treat cancer in model organisms such as mice, and then moved to humans to treat things like ovarian cancer, leukemia first. Leukemia. The only problem is it had some bad side effects. So not only did it kill cancer cells, it killed a lot of other cells too, which made the patients quite sick. So chemists went to the lab and they made a couple of different chemicals that are shown in the notes of your slide. So if you go down here, so this was camptothecin was discovered in the 1970s. And the chemists made less toxic forms of camptothecin called topotecan and irinotecan. Treats ovarian cancer, small cell lung cancer, and colon cancer. So it was an improvement and had less side effects. But how does it act? They didn't know at the time, but 20 years later after its discovery, they discovered of the drug. They discovered why the drug worked. And the chemical work, camptothecin, and what it does is it inhibits topoisomerase 1. So what does that have to do with replication? Well, topoisomerase 1 is part of the replication machinery. It's one of the proteins involved in DNA replication. And what it does is it releases the torsional stress. So as the DNA is unwound to let DNA polymerase in there and make new strands of DNA from the old strands, pulling apart that helix puts some pressure on the other regions of the DNA that haven't been opened up yet. And so either that's just going to break under the stress, or there's going to be a way to release that stress. And so the way to release that stress is for topoisomerase to chop the DNA, both strands of DNA, unwind the DNA to release that torsional stress, and then boop, put it back together. So that's what topoisomerase does. It relieves the torsional stress in DNA replication. And why does camptothecin, DNA replication has to happen in a lot of cells in your body. So why is it the camptothecin can work against cancer? Well, cancer cells are replicating at a higher rate. They're undergoing the cell cycle and cell division at a higher rate than most of the cells in your body. And so they are more sensitive to drugs that affect DNA replication. However, camptothecin is toxic for a similar reason. It's going to affect all of the cells in your body that undergo replication on a rapid rate. And that includes the lining of your digestive system, which is replaced pretty frequently. I think like every day to 3 days or so, like that. Because it's got a lot of food going through. It's going to slough off the upper layer of epidermal cells, and so your stem cells are going to be constantly creating new tissue for your whole digestive tract. So that's why a lot of cancer patients will lose their appetite, because those cells are going bye-bye in their digestive system. So there you go. Topoisomerase, DNA replication, and cancer. That's how they all fit together. Some student learning objectives to get you started. These will apply for Part 1 and Part 2. And then I'll have more student learning objectives when we get to Part 3. So this is for Part 2 right here. This is on polymerase chain reaction. So we got a little overview. let's see. I don't know if I can click to this. Oh yeah, I can. Cool. Overview of replication is here. Do I get sound?
The initiation of replication requires a particular DNA sequence called the origin. This sequence is called ORIC in the E. coli chromosome. A protein called the initiator protein binds to ORIC and causes the DNA to unwind. Unwinding requires several proteins. The most crucial are DNA helicase, which breaks the hydrogen bonds that hold the two DNA strands together. Single-strand binding proteins, which help maintain the DNA in the single-stranded state. And DNA gyrase, which reduces torsional strain.
That's also topoisomerase. DNA gyrase is what it's called in bacteria. Topoisomerase is what we use in eukaryotes.
An enzyme called primase synthesizes short RNA primers to allow DNA synthesis to begin. DNA polymerase III synthesizes DNA, beginning with the free 3' hydroxyl group on the last RNA nucleotide of the primer. Because the two nucleotide strands of DNA are anti-parallel, replication takes place continuously on one strand, the leading strand, and discontinuously on the other, the lagging strand. The short lengths of DNA produced by discontinuous replication of the lagging strand are called Okazaki fragments. DNA polymerase I cuts out the RNA primer and uses 5' to 3' polymerase activity to replace the RNA nucleotides with DNA nucleotides. DNA ligase seals the nick between the DNA added by DNA polymerase III and that added by DNA polymerase I without adding another nucleotide to the strand. Through these steps, the replication machinery of the cell uses the parental DNA as a template to synthesize the leading and lagging strands.
All right. So that's an overview of DNA replication. Genetic information must be accurately copied every time the cell divides. So replication has to be extremely accurate. One error in a million base pairs leads to 6,400 mistakes every time a cell divides, which would be catastrophic if we didn't have a way to correct those mistakes. Replication also takes place at high speed. For example, in the bacteria E. coli, DNA is replicated at a rate of 1,000 nucleotides per second. I ask you, can you write 1,000 letters on a piece of paper accurately in 1 second? Your answer is likely no, and neither can I. So it's happening at a very fast rate. So one safeguard for keeping accuracy during DNA replication is this idea of semi-conservative replication. It reduces the number of errors because the template DNA provides basically instructions. And there are 2 template strands in DNA replication. So how did we come up with this idea of semi-conservative replication? Well, there were 3 models of replication shown here: conservative replication, dispersive replication, and semi-conservative replication. I all start with original DNA labeled in blue, and then as we make new DNA, it is shown in red. For conservative replication, the model suggests that the original DNA stays intact and we get a completely new double-stranded DNA molecule, and then we continue to do that. So as you go through your levels of replication, you get more and more double-stranded new DNA, and you keep your fully conserved old DNA strand, double strand. Dispersive replication, you again start with that original DNA double strand in blue, and you break apart what we now know as the phosphodiester bonds and reassemble it just like a jigsaw puzzle. And so you continue to get old mixed with new for every subsequent DNA replication. So you have blocks of new and old mixed together. In semi-conservative replication, the old DNA double strand is separated, as Watson and Crick suggested, by breaking those hydrogen bonds and having one old DNA as a template for making new DNA. And so your second round, you're going to have a mix of old and new, and in the third round, the first round of replication, you have a mix of old and new, and the second round of replication, you've got some that are mixed old and new, and some that are 100% new. So these are the three models, and so there was a couple of scientists by the last names of Meselson and Stahl that said, hey, let's test those replication models and see which one is true. And they did that by using some heavy isotopes. These are not radioactive, but they're just heavy. So there was a rare isotope of nitrogen called nitrogen-15 that they used. And they used nitrogen-14, which is most abundant in our world. Of course, my question to you is, what portion of DNA contains nitrogen? And you'll likely respond, well, Dr. Silverkey, of course, it's the nitrogenous bases. And so here we are looking at cytosine labeled with either nitrogen-14 or nitrogen-15. Nitrogen-15 being heavier than nitrogen-14. So when DNA is made of nitrogen-14 and 15 and you put it into a test tube with a semi-solid matrix, well, anyway, it's a liquid that's a little bit like gelatin, and you spin it in a centrifuge, you get centrifugal force. The things that are heavier are going to travel the farthest in this gel-like substance, gelatin-like substance. And so you can see that DNA labeled with the heavier isotope N15 is going to travel further down the test tube. You know, Newton's law of motion, things that stay in motion tend to stay in motion, especially if they're really heavy and it's hard to stop them. Or DNA with N14, which is less heavy, is going to migrate a smaller distance in this gelatin-like substance that's found in the test tubes, the centrifuge tubes. So this is what they did. They grew up some lovely bacteria, our friend the E. coli from your gut again, in the presence of nitrogen-15. Okay. And they would slice open those cells, look at the DNA, spin it in the centrifuge tube, and they found where the original DNA would settle was in a band that was really far down in the centrifuge tube, shown in blue. Then they saved some of these bacteria. They didn't lyse all of them. They grew them in N15. They spun them down, took off the liquid, and then suspended them in nitrogen-14 containing media and let them replicate. And what they found was not, and when they spun it, when they broke open the cells, got the DNA out and spun it in this gelatin-like substance in the centrifuge, they found that they lost their heavy DNA and they had something that was a little bit lighter. This, if you look at the models, this is going to eliminate this first conservative replication model because they would have kept their old, fully conserved original DNA and they just lost it in the second, or the first round of replication right here. Okay, so we got our control, got our first generation. Then they, spun these down, took off the nitrogen-14, transferred these bacteria to a new flask with more media. It has the sugar and all the good food for the bacteria to grow. That had N14, nitrogen-14 in it. They stopped growth. they broke open the cells and took out the DNA, and they spun it, and they found that they retained this band here that's a mix of old and new material, and now they have appearing a lighter band made of 100% nitrogen 14. So that eliminates dispersive, because dispersive never, it just has a band, one band all the time. First it was really heavy, then it was lighter, a mixture, and then it was going to be even lighter, so it was going to go higher and higher in the centrifuge tube. So what's left is semi-conservative. We've got some brand new stuff made of only nitrogen 14, and we have some hybrid stuff that's made part of nitrogen 14 and part of nitrogen 15 in all the subsequent rounds of the experiment. And so just in summary, the controls are all nitrogen 15, are grown, if the bacteria is grown in just nitrogen 15, you get a really heavy band. If cells are grown in nitrogen 14 only, you get a really light band. And if you grow them in nitrogen 15 first and then transfer them to nitrogen 14, you're going to get a hybrid band that's half nitrogen 14, half nitrogen 15. And if you take these bacteria and do one more round of replication, you're going to keep that hybrid band, and you're now going to have a band that's made 100% of light nitrogen 14. So you should be able to recognize and explain, these results on your exam. How many bands will you see, type thing, depending on the experiment. So the N15, N14 experiment supported the hypothesis of semi-conservative replication. So here's our concept check. How many bands of DNA would be expected in Meselson and Stahl's experiment after 2 rounds of conservative replication? So you guys should try to figure that out on your own. You can pause the video and try to figure that out. The answer is you're going to have 2 bands. One is going to be fully conserved nitrogen 15 band. The other one is going to be nitrogen 14 created band. So if I were to draw that, it would look something like this. So I've got my test tube. Use my finger, and I've got my nitrogen 15 band, and then I've got my lighter band up here, which is nitrogen 14, and this would be conservative replication. Conservative. Okay. So you should know what the results would look like if it was dispersive. If it's dispersive, you're always going to have a band that's, well, you're going to have, originally you're going to have like this. And then the next round, whoops, you're going to have something that's mixed, right? And then in the next round after that, it's going to be even higher, because you're going to get more and more new material added to it. So this would be an example of dispersive, if that's what we're happening. But we know that neither one of those is happening. So back to the PowerPoint. Replication begins at origins of replication that we can call replicons. And this is a table 12.1 that looks at replicons. In other words, the replication origins or origins of replication that are found in different species. If you're a bacterial cell like E. coli, Escherichia coli, you've only got a single circular chromosome. A single circular chromosome. So it's going to have a number. How many replication origins is it going to have? Just one. That's all it needs. Okay. This is the length of the DNA. That's not the number of replication organs. This is the length of how much DNA is going to be replicated. See, this is supposed to be length. So length of replication origins. In a yeast, we've got more than one chromosome. So you're going to actually have 500 replication origins. You can see we get Drosophila and fruit fly is Drosophila. Xenopus is frog, and musculus is mouse. As you increase in complexity of the species, you're going to have more and more chromosomes, and you're going to have larger amounts of DNA. So your replication origins are going to increase, the number of them. But the lengths of DNA that being replicated doesn't necessarily increase. Well, it does for frogs and so forth. But look, compared to bacteria, these numbers are a lot lower. So even though we've got more DNA, the cell is using more origins of replication, so we can replicate all of this large amount of DNA in a shorter period of time. There are 3 modes of replication. They're called theta, rolling circle, and linear. The first 2 are found in bacteria. You should study table 12.2 so that you know what's happening in each one. So your DNA template is circular in the first 2 and linear in the third. The only one that we break the nucleotide strand in is rolling circle. It's going to tell you the number of replication origins or replicons, one versus many, and whether it's unidirectional or bidirectional, and how many products you're going to get and what shape they're going to be. Are they going to be circular or are they going to be linear? So make sure you study table 12.2 and know it. This is theta replication right here, and it's named after the Greek letter theta because John Kane, who took these pictures you see down here in 1963, excuse me, noticed that right here, the DNA under the electron micrograph looked like the Greek letter theta. And it's kind of hard to see here, but it's easier to see here in the cartoon picture. And so we have an origin of replication that opens up. The new DNA is shown in red. The old DNA is shown in gray. And the replication bubble gets bigger and bigger until we're done with replication. And it's just at this one point where it looks like the Greek letter theta. So what results is we get 2 circular DNA molecules. And rolling circle replication is a bit different. We have the single circular chromosome, but we get it broken here. And then we start making the DNA shown in red. And as we make the new DNA, we push one of the 2 old strands off. And it can actually go off on its own and recircularize and be a template for new DNA strand so that you can end up with double-stranded DNA. I want to go over to the whiteboard here in a second. So we can actually transfer antibiotic resistance genes from one bacteria to another using this method of rolling circle replication. So I'm going to open up another page of my whiteboard to illustrate that. All right, so if I have a bacterial cell shown here that has what we call an F pilus, And inside we have our old DNA. And one old DNA is getting pushed off as we make our new DNA. This old DNA can go into a new bacteria cell right here that has its own double-stranded DNA molecule. But we can get this in here and it can circularize and then be a template for DNA. And you know, this one can actually be smaller and can be what we call plasmid DNA. And it can carry a gene on it that we got from the original bacterial cell that can give you resistance. So you can resist antibiotics. Sorry, these are overlapping. So this is what we call horizontal transfer or horizontal inheritance. So these can both be parental cells, and you're passing DNA from one parental cell to another, and it's carrying something that can give it a survival advantage. And this is how we get multi-drug resistant bacteria such as MRSA in our hospitals, because we have a lot of antibiotics there, and the bacteria can do this action right here. And just to let you know, this is in contrast to what we call vertical. Oh dear, I gotta get a pen. Vertical transfer is what we've been studying before, where we've got things like binary fission and things like mitosis, is where we have vertical transfer or inheritance of DNA. Into the daughter cells. Of course, that's with eukaryotes. And we're going to get identical daughter cells from our parent cell. All right, going back to the PowerPoint. The last mode of replication is linear eukaryotic replication. And it's shown here. It has multiple origins of replication, unlike the other two modes, which only have one origin of replication. And DNA replication goes in both directions, therefore it's bidirectional. And these bubbles, these replication bubbles, just keep getting bigger and bigger until they run into the other replication bubbles, and you have complete DNA synthesis. So each replication bubble has what we call two forks. So there's a fork on each side of the replication bubble. And of course you have multiple replicons or origins of replication on your chromosomes, your linear chromosomes. You have 20,000 to 300, that they can be as big as 20,000 to 300,000 base pairs in length. Principles of replication. Replication is always semi-conservative. Replication begins at sequences called origins of replication, which are AT-rich because they only have 2 hydrogen bonds in between them, so they're easy to break. Genomic replication occurs once per cell cycle, and in eukaryotes this is regulated by a protein called replication licensing factor. Okay, it's going to stop the cell from replicating DNA more than once per cell cycle. Remember that. It's only in eukaryotes. Central problem with replication is that it requires rapid DNA synthesis. Large amounts of info are copied. Errors can happen spontaneously. So what do we do about that? How does it remain essentially unchanged? Well, factors that reduce it are semi-conservative replication, then a function called exonuclease that we're about to talk about with DNA polymerase. And if any of these have failed, then we've got DNA repair mechanisms. The hydroxyl group, so DNA is replicated in the 5' to 3' direction. The hydroxyl group off the 3' carbon attacks the phosphate on the 5' carbon thusly. So we got our extra electrons on our hydroxyl group, and they're going to attack the phosphate group. The two inorganic phosphates leaving you with a phosphodiester bond. And then we have a new 3' end that can attack the next phosphate. And this is shown in this animation, which I'm going to show part of.
DNA polymerases catalyze the synthesis of new DNA strands from a DNA template. DNA polymerases require a pre-existing RNA or DNA strand, the primer, to initiate new DNA synthesis. These polymerases add deoxyribonucleotides only to the 3' end of a growing strand. Taking a closer look at the nucleotide polymerization reaction, the 3' end of the primer contains a free 3' hydroxyl group. The 3' hydroxyl reacts with the 5' end of the next free nucleotide to be added.
Okay, I'm going to stop it there. I'm running out of time on this video. I might have to have this trickle over into a second one. But we have a 3' hydroxyl to start the synthesis. An enzyme called primase makes an RNA that provides that 3' hydroxyl. And here's an RNA primer right here with the 3' hydroxyl. In bacteria, like E. coli, DNA polymerase 3 is the first enzyme that comes along and recognizes the primer and starts synthesizing DNA in a 5' to 3' direction using its polymerase function. But it can make a mistake where you've got a C across from an A, and then it has to use its exonuclease function, which goes backwards 3' to 5' to remove that mistake, and then continue synthesis to create the repaired DNA. Then DNA polymerase 1 comes in, and it has 5' to 3' exonuclease function to remove that RNA primer and simultaneously put in new DNA. Then finally DNA ligase comes in and seals the nick together. And you can study this in this table 12.3. You should know who has polymerase, 3' exonuclease, and 5' to 3' exonuclease activity in DNA polymerase 1 and DNA polymerase 3.